Chapter 9 Medical Mycology Outline Introduction Morphology of fungus, fungal reproduction, fungal classification, fungal ecology and metabolism, fungal infection, mycosis, laboratory diagnosis of fungal infections, antifungal drugs. Objective: at the end of this session, you have to know what is mycology and what it concerns with the way of reproduction, the classification of fungi, what is the fungal ecology and the metabolism, disease caused by fungi, laboratory diagnosis and available antifungi drugs. Mycology. Greek word myx, mushroom, the study of fungi. Considered a botanical science. Medical mycology, the study of fungi that are associated with human disease. Diverged from botanical mycology around 1920s. Book on dermatophytic fungi, published by Raymond Savo Roud. Fungi have become public health hazard. Increasing size of population at risk. Transplant recipients, critical care patients, aging population with debilitation. General characteristics. Kingdom fungi, eukaryotic, membrane-bound cell organelles, simpler in structure, heterotrophic, lack chlorophyll, embedded in a food source or medium and secrete enzymes for external digestion and absorption of nutrients. Most fungi are obligate for facultative aerobes. Cell membrane contains ergosterol rather than cholesterol. Most species are beneficial to humankind, breaking down and recycling organic matter, production of food and alcoholic drinks, including cheese, bread, and beer. Served medicine by providing useful bioactive secondary metabolites such as antibiotics. Example, penicillin, cyclosporin, as a model systems for the investigation of a variety of eukaryotic processes. Medically important fungi exist in two morphologically forms, mold or yeast. Molds, filamentous, multicellular forms mostly found outside the body and at room temperature as growing organisms on fruit, bread and cheese. Hypha is the basic microscopic unit of the molds, longitube-like structure, responsible for the fluffy or cotton ball appearance of the macroscopic mold colony. Molds are classified and identified partially on the basis of whether the hyphae are septate or aseptate. Septate hyphae, filaments with cross walls or septa that partition the hyphae into individual cellular compartments. Pores, pores that allow the migration of cytoplasm and many organelles. Accepted hyphae. Accepted hyphae not divided by septa are coinocytic or multinucleated. Hyphae grow as an intertwined mass of filaments, collectively called mycelium. When mold grow on substrate, it can form vegetative mycelium or thallus, nutrient-absorbing and water-exchanging portion of the mold grows in or on a substrate, is actively growing and forms the main body of the colony. Aerial mycelium, fungal filaments or hyphae that protrude above the surface. Yeasts are single cell, usually spherical to ellipsoid in shape and occurs singly in pair or form a chain or bud structure mostly found inside tissue or at body temperature. Dimorphism. Some fungi grow only in the yeast form, others grow only as molds, but there are fungus that exist as mold and yeast depending on certain growth conditions. Such phenomenon is dimorphism. For example, many fungi grow as molds at room temperature 24 degrees centigrade but transforms into yeasts at human body temperature 37 degrees centigrade. Most of the more dangerous human pathogenic fungi are dimorphic. Fungal reproduction. Sporulation, process of making spores, principal means by which fungi reproduce and spread through the environment. Fungal spores are metabolically dormant, protected cells, can be borne by air or water to new sites, where they germinate and establish colonies. Generated either asexually or sexually. Molds reproduced by asexual spores and the yeasts reproduced by budding and sexual sporulation. Many fungi can produce both sexual and asexual spores. Fungi that exhibit sexual phase, teleomorph, are known as perfect fungi. Fungi for which the sexual cycle is either unknown or is missing entirely are called imperfect fungi. Sexual spores are rarely agents of human disease, except for the deuteromycetes. All fungi produce sexual spores. Asexual spores are generally responsible for initiating human infection. Fungi classification. Kingdom fungi classified into 5 divisions based on spore types, morphology of hyphae, sexual cycles. These divisions are Chytridiomycota or Omycota, Zygomycota, Ascomycota, Basidiomycota, Deuteriomycota. Fungal ecology and metabolism. All fungi are carbon heterotrophs, depend on exogenous nutrients for carbon and energy. Most fungi are obligate or facultative aerobes. Many species are capable of maintaining metabolic activity in basic nutrient mediums. The unknown metabolic types of fungi include thermophilic, psychrophilic, acidophilic, and halophilic species. Fungal ecology. The metabolic capabilities of fungi are exploited in production of bread, wine, beer, cheese. Pharmaceutical industry. Example is the production of antibiotic substance, enzymes, citric acid, etc. The metabolic activity of fungi can also be a damaging factor. Fungal infestation can destroy foods, wooden structures, textiles, etc. Fungi also cause numerous plant diseases, in particular diseases of crops. Fungal ecology. Based on their metabolism, saprophytic fungi, free living organisms, obtain nutrients from dead organic matter. Important decomposers in geochemical cycles. Mutualistic fungi, relationships with plants or microbes in which both partners benefit. Parasitic fungi acquire nutrients by attacking live animals or plants. Fungal infections, mycosis. Diseases caused by fungi are collectively called mycosis. Singular mycosis. Pathogenic fungi may be grouped into true pathogenic and opportunistic pathogen fungi. There are 4 general categories on the basis of the primary tissue affinity of the pathogen. 1. Superficial mycosis. 2. Cutaneous mycosis. 3. Subcutaneous mycosis. 4. Systemic mycosis. Superficial mycosis are fungal infections confined to the outermost dead layers of skin, hair, and nails. Symptoms: discoloration, scaling, or depigmentation of the skin do not elicit a cellular response from the host. Infections are generally painless. Commonly known superficial fungal infections include Tinea versicolor, Tinea nigra palmaris, Tinea pedra, Tinea versicolor, Pityriasis versicolor. Tinea versicolor caused by Malassezia furfur is a chronic asymptomatic superficial fungal disease characterized by fawn to brownish colored desquamating macules involving principally the trunk. The infection is often asymptomatic and may not be noticed by the patient, but sometimes a slight pruritus may develop. This diagram shows versicolor. Tinea nigra palmaris. Tinea nigra caused by Exophilia wemekii is tropical asymptomatic fungal infection usually involves the palms of the hand. The infection is asymptomatic, appearing as discrete pigment areas which have been said to resemble a skin stained with silver nitrate. There is no erythema or inflammatory reaction. Tinea pedra. Pedra is a fungal infection of the hair characterized by the presence of stony, hard nodules along the hair shaft, caused by Pedraea horte, black pedra, trichosporon, beigeli, white pedra, which is limited to the hairs of the scalp, beard, and mustache, causes no discomfort to patients since only the shafts of shafts of the hair are involved. Cutaneous mycosis.